To study the cellular components, to elucidate their biochemical function and structure, we must first separate them by their physical properties with a technique called fractionation. Fractionation led to monumental discoveries in the function of the mitochondria, endoplasmic reticulum, and Golgi. We will discuss fractionation in the context of Kornberg, Lehmann, Bestmann, and Sims's experiment to purify DNA polymerase I. The first step in fractionation is breaking the cell with detergent, ultrasonic vibration, osmotic shock, and/or a blender. The resulting slurry is called homogenate. Step 2 of fractionation is ultracentrifugation. Centrifuging at low speed, the mixture separates into two layers, one supernatant, one pellet. The first pellet contains the nuclei and cytoskeleton. The supernatant, containing everything else, is pipetted into a new tube and subjected to medium-speed centrifugation. The second pellet contains mitochondria, lysosomes, and peroxisomes. High speed forms a third pellet with even lighter components, microsomes and small vesicles. At very high speeds, we can get a fourth pellet containing ribosomes, viruses, and large macromolecules. The polymerase discovery team worked with an extract of E. coli, phosphate tag DTTP, magnesium ions ATP, and buffer. This was the 1950s, 20 years prior to the invention of sequencing, so the scientists did not have much information to work with. They expected the polymerase to be a protein. Proteins can be separated by molecular weight, charge, pI, hydrophobicity, solubility, and binding affinity. Kornberg and co. added streptomycin sulfate to precipitate all nucleic acids in a p fraction. The scientists used ammonium sulfate, C-gamma, and acetone fractionations in combination with ion exchange chromatography. For chromatography, start off by clamping the column to a retort stand at 0.5 milliliters of water, mark the column, and let the water drain. Kornberg and co.'s experiments used diethylaminoethyl cellulose and phosphocellulose, abbreviated DEAEC, as their positively charged resin. DEAEC is an anion exchanger. It can bind to the negatively charged phosphate component of DNA. Buffer is added to equilibrate the column. The sample containing DNA polymerase is added to the column. DNA polymerase is bound to the resin. It will not elute in the flow through fractions. More buffer is added to wash away everything besides the target protein. To finally separate DNA from the resin bed, a salt such as NaCl must outcompete the interaction. The eluate fractions contain DNA polymerase. With that, the fractionation process is complete. To review, Step 1, break the cell membrane to form homogenate. Step 2, separate organelles by size and density. Step 3, apply a combination of fractionation techniques, usually including column chromatography. It is this process that has led to our current day understanding of cells. Thank you.